So now we'll move on to our next speaker. Our next speaker is Dr. Julie St. Pierre from the University of the University of Ottawa. She's the director of the Metabolics Core Facility, and she holds a CRC Tier 1 in cancer metabolism and is co-leading a Terry Fox program project grant on oncometabolism. The central research focus of Dr. St. Pierre's laboratory is the understanding of metabolic adaptations in cancer. During the last decade, her team contributed to understanding the role of metabolic regulators in cancer with a particular focus on poor outcomes in breast cancer. They showed that PGC1 alpha plays a key role in setting the metabolic state of poor outcome breast cancers, and then it promotes breast cancer growth and metastasis. Recently, they are pursuing research projects on metabolic adaptations, fueling metastasis and therapeutics resistance. Now we'll hear from Dr. Julie St. Pierre on her work in her lab.
All right. So, yeah, first I want to thank Anna and the organizing committee for this wonderful conference and the opportunity to present my work here. So today I'm going to talk about metabolic plasticity in breast cancer progression, and specifically what I'm going to discuss is our attempt to find metabolic vulnerabilities in therapy-resistant breast cancer. All right. Okay, I'm having problem with this slide. Okay, so when we think of cancer and metabolism, I have a very metabolic-centric point of view about metabolism in cancer. So if we start from a normal cell that you could see here to a transformed cell, as you know, you can get oncogene activation or tumor suppressor inactivation. But along with that come metabolic transformation. And this is really what we're interested in. And we really want to identify the metabolic dependencies of cancer so that we can target this therapeutically. Okay. So in terms of metabolic adaptation in cancer, probably the most well-known adaptation is called the Warburg effect. So it's called the Warburg effect because it was discovered by Otto Warburg in the 1920s. And what Dr. Warburg found was that if you take tumors, even in the presence of oxygen, they actually use glycolysis to generate ATP instead of burning glucose in their mitochondria. So when you use glycolysis instead of mitochondria, you only get 2 ATP per unit of glucose instead of 36. So in order to maintain ATP balance inside cancer cells, what they do is they uptake a lot of glucose. And this dependency of cancer cell on glucose is actually used in the clinic, where we can image tumor using an analog of glucose called fluorodeoxyglucose. So this is what you can see here. So these cancer cells, they take up a lot of glucose to make up for the fact that they only get 2 ATP when using glycolysis instead of using their mitochondria and giving 36 ATP. But I want to point out that not all cancer cells are like that, and not all cancer types display the Warburg effect, although many do. And in general, it's thought that cancers that are aggressive display the Warburg effect. So in my lab, we're looking and studying advanced breast cancer, and I'm just showing here our general workflow. So we use multi-omic approaches. So we start with expression profiling. So we do transcriptomic analysis. Given that many transcription factors regulate the metabolic state of cells, we also do chip sequencing, where we can actually see on the promoters of which gene-specific transcription factors are sitting. So this is our transcriptomic workflow. And then we couple with that, we do metabolomic analysis. And our entry into metabolomics is always doing bioenergetic profiling. So we actually quantify how these cells are getting their ATP. Is it through glycolysis, mitochondria, both? And we can quantify the proportion. And then we do metabolite profiling using metabolomics. And then, of course, we need great bioinformatics to actually integrate all these data sets. So as I've mentioned, today I'm going to talk about therapy-resistant breast cancer, which is a real challenge clinically. So this slide here, I'm just showing different therapies that are currently used in the clinic. So you could have specific tyrosine kinase inhibitor, for example, lapatinib, that is used in HER2-positive breast cancer. There's some chemotherapeutic that's used, for example, taxane, like paclitaxel and docetaxel is being used, and also anthracycline, like doxorubicin and epirubicin. And of course, with treatment, often cancer cells develop resistance. There are various ways by which they can do this. They can actually upregulate some ABC transporter and export the drug out of the cell such that the concentration inside the cell is lower. Sometimes they metabolize these drugs into less toxic compound. For example, like for the anthracycline, you could have the alpha-ketoreductase that actually make the anthracycline less toxic. And also sometimes what you have is that those drugs are sequestered inside lysosomes so that they can cause less problems for the cells. So when we embarked on that project, we were examining resistance to anthracycline, specifically doxorubicin and epirubicin. So to study therapy-resistant breast cancer, we modeled that in MCF breast cancer cells, and it takes a long time to generate these models. So these therapy-resistant models were generated in the laboratory of Dr. Amadeo Parisenti, where you actually expose breast cancer cells to increasing concentration of epirubicin and doxorubicin, and it takes several months to a year. And then up until you have resistant cells. So this graph here on the right, what you see is that if you take, sensitive MCF breast cancer cell and you expose them to doxorubicin and epirubicin, they cannot grow, while the resistant cells are still able to grow, just showing that indeed these cells are resistant to these anthracycline. And then to come back to our experimental workflow, we did transcriptomic and metabolomic analysis to understand, are there similarities and differences for breast cancer cells that are resistant to doxorubicin and epirubicin. So we did gene expression profiling. So this is a principal component analysis graph. And even though doxorubicin and epirubicin are both anthracycline, like if you do their transcriptomic profile, you can separate them nicely on the principal component analysis graph. And they also separate nicely when you do the same analysis using metabolomic data set for these resistant cell lines. So when we look a bit further into the transcriptomic data set, not surprisingly, many of the biological pathways that were found to be modified in these resistant cells were common between doxorubicin and epirubicin resistant cells. But we wanted to focus our analysis on potential differences between these two anthracycline. So what we observe is that, for example, if we look here for the DUX resistant cells, they seem to be upregulating glutathione metabolism in contrast to epirubicin resistant cells. And when we look at epirubicin resistant cell, they had a tendency to increase OXPHOS compared to the doxorubicin resistant cells. So completely separate from this transcriptomic and metabolomic analysis, we also perform a targeted shRNA screen using doxorubicin resistant cells and epirubicin resistant cells. And what's great about doing an shRNA screen when you have these resistant cells is that these cells always grow in the presence of the drug that they're resistant to. So when you come with an shRNA, if the cell become more sensitive, you're actually losing these cells in the screen. So you're looking for, which cells you're losing over time when you come with those shRNA. And what we found is that for the epirubicin resistant cells, again, oxidative phosphorylation was coming as a strong hit. And for the doxorubicin resistant cells, we had glutathione metabolism coming as a strong hit. So this is a completely separate systemic analysis, like we did the transcriptomic and metabolomic, and this was an independent shRNA screen that, all these data were pointing towards the same direction, that OXPHOS appears to be quite important for epirubicin resistance, while glutathione metabolism seems to be quite key for doxorubicin resistance. So we then did some mechanistic work. We wanted to see, okay, so if the doxorubicin resistant cells depend on glutathione, maybe we could trace. So we used glutamine that was labeled, and we look how much of this glutamine is used to generate reduce and oxidize glutathione. So we did that in the control cells, the doxorubicin resistant cells, and the epirubicin resistant cells. And what you can appreciate here on the graph is that as you can see with the green bars, like the generation of glutathione was much increased in the doxorubicin resistant cells compared to the epirubicin resistant cells under control. So showing really that these cells use that pathway more significantly than the other cells. Then, of course, if you have more or you generate more glutathione, it's likely to be used, to respond to oxidative stress. So we also look at the expression of genes that are involved in the detoxification of reactive oxygen species. And we said that many of these genes, their expression was increased in doxorubicin resistant cells. Some of them were also increased in epirubicin resistant cells, but not to the same extent as for the DUX resistant cells. And then when we look for the production of reactive oxygen species at baseline, we could see that there was less in the DUX resistant cells than the EP resistant cells. And if we challenge these cells with a Ross stressor, again, the DUX resistant cells were doing better than the epirubicin resistant cells. So I've shown you so far that doxorubicin resistant cells depend on glutathione metabolism for resistance. Now we're going to look at epirubicin resistance. As you may recall, like the transcriptomic analysis and the shRNA screen had pointed towards OXPHOS as being important. And one key regulator of OXPHOS is PGC1 alpha. And what you could see here and you can appreciate is that the epirubicin resistant cells have greatly increased expression of PGC1 alpha. And along with that, we're not going to go through all the data, but if you look at oxygen consumption rate, this is the epirubicin resistant cells, you could see that respiration is increased, both coupled and uncoupled respiration compared to DUX and control. So this is at baseline. And then if you look like, their maximal ability to actually use OXPHOS, the epirubicin resistant cells are actually have greater capacity to do that than the doxorubicin resistant cells. So again, showing functionally that, yes, these epirubicin resistant cells are dependent on OXPHOS. So we wanted to test if we could interfere with these pathways. So again, the goal of our research program is to identify metabolic vulnerability so that we could target these to help in the treatment of cancer. So far I've shown you that the DUX resistant cells are dependent on glutathione and the epirubicin resistant cells are dependent on OXPHOS. So what we did to target these cells is for the epirubicin resistant cells, because they're dependent on OXPHOS, we use metformin, which is a drug that's used for type 2 diabetes, but that is actually targeting complex 1 in the electron transport chain. And what you can appreciate when you increase the concentration of metformin, you could see that the epirubicin resistant cells are growing less and that the doxorubicin resistant cells are almost unaffected by this compound. Then if we look for the doxorubicin resistant cell, I've shown you that they're dependent on glutathione metabolism. So to interfere with that, we use BSO. And what you can appreciate is that if you treat the epirubicin and doxorubicin resistant cells with BSO, the doxorubicin resistant cells are extremely sensitive to that compound, whereas the epirubicin resistant cells are much less sensitive. And then we took this and tested this notion in vivo. So what we did is we used immunocompromised mice, and on the left flank, we injected the doxorubicin resistant cells, and on the right flank, the epirubicin resistant cells, and we treated these mice with BSO. And what you can appreciate is that the DUX-resistant tumors were more sensitive to BSO, so this is what's graphed here, whereas the epirubicin-resistant cells show very little sensitivity to BSO. So the take-home message from that story is that breast cancer that's resistant to doxorubicin is very dependent on glutamine and glutathione for growth, and coming with BSO can actually significantly limit tumor growth. Whereas if we take breast cancer cell that's resistant to epirubicin, they're very dependent on OXPHOS for their growth. And if you come with compounds like metformin that can block complex 1, you can limit their growth potential. So this was the first part. For the second part of the talk, I want to put those results and put them in context of a solid tumor and how therapy resistance can develop and evolve. So if we take solid tumor, so this would be the tumor here, and here you have the blood vessel. What you can appreciate is as you go towards the core of the tumor, there's decreased oxygen tension, there's decreased nutrient and energy, And also there's decreased drug concentration. And we wanted to investigate how this gradient of drug concentration in solid tumor can actually impact the development of therapy resistance. So this is just an example here where you can actually see, like inside a mammary tumor, so you have doxorubicin is shown in blue, and the blood vessels are shown in red, and the hypoxic region are shown in green. What you can see is that the concentration of doxorubicin in mammary tumor is highest when it's close to the blood vessel. And then as you move further away in the hypoxic region, there's very little doxorubicin. So I'm just showing a schematic here to represent what we're trying to understand is that there's a blood vessel here, and when you have a solid tumor, you'll have a concentration, drug concentration that is going to decrease towards the core of the tumor. And what you predict would happen is that in a solid tumor, those cells that are very close to the blood vessel and get exposed to very high concentration of chemotherapeutic would die. But then these cells would have been exposed to low doses of these chemotherapeutic, and gradually they would get exposed to increasing concentration. So this is what's shown here. As treatment progresses, like these cells at the beginning will get exposed to very low dose of chemotherapeutic, but as these cells die, these cells will get exposed to higher concentration. So this is just to show you, like coming back to the first story. So the first story was really focused on comparing therapy resistant or therapy sensitive cells versus therapy resistant cells. And in this story, we're really trying to understand the evolution of therapy resistance. So again, we took these cells, we had doxorubicin and epirubicin resistant cells. So these cells were exposed to increasing concentration of drugs. And we have cells that are resistant at different concentrations. So we have cells that are resistant to 1, 5, 10, 15, 25, 50, and 100 nanomolar doxorubicin and epirubicin. And then what we did, we used the same workflow that I've shown at the beginning. We do transcriptomic analysis as well as metabolomic analysis. We do pathway identification. And then the idea again is to find functional target that we could hopefully target therapeutically. And so this first slide here is our data, actually proof of concept, showing that if cancer cells are exposed to low dose of chemotherapeutic, and then suddenly they get exposed to increasing concentration as treatment progresses, you actually make them resistant to therapy. So we're going to look, for example, we're going to look for doxorubicin as an example. So what we did in that experiment is we take our sensitive cells and we mix them with cells that are resistant to 1 nanomolar, and then we challenge them with the next concentration, which is 5 nanomolar. Then we take the cells that are resistant to 1 nanomolar and 5 nanomolar, and then we expose them to 10. And we're looking which population is doing better. And as expected, if we look here in the middle, if we take cells resistant to 5 and 10 and we expose them to 15, the cells that had seen 10 before do better when they're exposed to 15. Similarly here, if we have cells resistant to 10 and 15 and they're exposed to 25, the 15 are doing better. And this is true for all DUX-resistant cells, and this is true for epirubicin-resistant cells. So if cells get acutely exposed to a very high concentration, normally they die or they stop proliferating. But if they're exposed to lower concentration and gradually they get exposed to an increasing concentration, this promotes resistance. So at the bottom here, what we're showing is actually the opposite of this, is that we take all the cells that are resistant to different concentration and we expose them to the maximal dose. And what you can see is that the cells that are resistant, the sensitive one, and those that are resistant to 1, 5, 10, 15, when they're exposed to 100, they don't do very well. But those that had seen 25 and 50 do much better. Again, showing that it's important to come fast with a high concentration, and that exposure to lower concentration actually promotes resistance. Then we did transcriptomic analysis, intracellular metabolomic analysis, and extracellular, so looking at the cell media metabolomic analysis for all DUX and epirubicin-resistant cells. And the point I want to show on this slide is that the cells that are resistant, or the sensitive cells and those that are resistant to low concentration, tend to cluster together. And then here, resistance increases, and these are the cells resistant to very high concentration. So you can divide like the data in kind of 3 blocks. You have the cells that are resistant to low concentration, middle concentration, and high concentration tend to cluster together, both in terms of transcriptomic and metabolomics. What we're showing here is that we look at the pattern of all genes in the transcriptomic data set, and then we let them see, what kind of pattern they develop, like with increasing concentration. So here on the left, you would have sensitive cells, and on the right, cells resistant to 100, and the whole dynamics in between. So as you can see, if you take all genes, some genes have a linear pattern with development of resistance. So for example, you could take this pattern here, it's going down. So these are genes that are involved in metabolism. You have some genes that, are upregulated early. We have some genes that follow a transient pattern like this. So they go up and then down when they reach high resistance. Some other genes follow another pattern. So some would go down and then up later on. And then we have some genes that have a late expression pattern. So they're pretty constant throughout. And then at the end, when the cells reach high level of resistance, their expression actually drop. So then when these data get published, everyone can look at their favorite gene or favorite pathway and see like how do they evolve with therapy resistance. So what we tend to find is that, here those genes involved in metabolism tend to go down. And then we have many genes involved in immune response and lysosome going up as therapy resistance develop. In terms of metabolomics, so we did similar thing, and we look at all metabolites that we could identify, and we made, the pattern with resistance. So again, sensitive cells on the left, fully resistant on the right. So some metabolites show a linear response with resistance. Some respond early. Some have a transient response. And what was very striking for the doxorubicin resistant cells is that we had many metabolites that actually were drastically decreasing when the cells acquired like high levels of resistance. So for example, like if you take C4 here, so we had many amino acid going down. And then for example, pyrimidine, which is obviously important for cell cycle. So these were going down at high levels of resistance. Then we did similar analysis as we just did for the DUX resistant cells for the epiribicin resistant cells. And again, if we look at the transcriptomic data, we had genes showing a linear pattern. We had some showing early response. That's what we saw. We had also the same transient pattern, some going up and down or some going down and up. And then we had the late pathways that were actually stable throughout. And then at the end, when cells were acquiring high levels of resistance, it could go down. So again, as I said, when these data will be published, everyone can mine their favorite pathway or favorite gene and see how they track with, epiribicin resistance. So we had different genes, but globally, like we had some similar patterns to what we found with doxorubicin, whereas, lysosome was going up with resistance, and we had some immune regulated genes that came on and were important. In terms of metabolites, we mostly found 3 patterns with resistance. So we had linear, early, and transitory. And then again, we had like purine and pyrimidine metabolism, as well as amino acid coming up as being very important. So obviously, there's a lot of data. I cannot present all this today, but what we did in terms of, I'm going to present what we did with the transcriptomic analysis, and now we're finalizing like our targets for the metabolomics analysis. But for the transcriptomic analysis, what we reason is that, we had some gene that show linear increase or decrease with resistance, some had transient pattern, as you saw, some had early response or late response. But the goal really therapeutically is to be able to come and hit a target that will sensitize cells regardless of where they're located inside a solid tumor. So we want to be able to target cells that are very close to a blood vessel and see high concentration of drug, as well as cells that are in the core of the tumor that, they're probably hypoxic and they don't get exposed to a lot of drugs at the beginning. So we decided to focus on those genes that had their expression was linearly correlated with resistance, both for doxorubicin and epiribicin. And then what we did once we identified these genes, so we had, I'm just showing you here a few examples. So we had BRWD1, SRC1, and ENDO-G. So BRWD1 is important for chromatin remodeling. SRC1 is an inhibitor of SRC, and ENDO-G is an mitochondrial endonuclease. So what you can see for the first two, their expression is increasing with resistance. So this is what you can see nicely here for DOX and AP. And then the expression of ENDO-G was actually going down with resistance. And then what we did is we took those genes and mined, publicly available data set for breast cancer patient. And what you can see is that the expression of BRWD1 and SRC1 is actually tracking with poorer outcome in patient. So this is what you see here for BRWD1. So when you have high expression, you have poorer survival. It's the same for SRC1. And then for ENDO-G, what we see is that it's going down. And what you can appreciate is that when you have low expression, is also tracking with lower survival in patient. So then what we did is we took all our cell lines that were resistant to DOX and AP, and then we knocked down those genes. So I'm showing here for SRC1. So at the bottom here, so it's shown very small. So we actually knocked down the expression, and what you could see is that the growth of these cells, regardless of their level of resistance, you could limit actually their growth. So we identify some target that regardless of where those cells would be located inside a tumor, they would actually be responsive to the inhibition of that gene. So we're doing the same now for the metabolomic data set. So we identify some important SLC transporter that could be targeted. So we're finalizing these analyses. So the take-home message for the talk is really what we're trying to do is we're trying to target and identify first metabolic vulnerabilities, and then to target these for therapeutic purposes. Of course, cells, they always have trick and they have a way to escape this. And what I want to end the talk on is really what's important is when you have this knowledge, you can anticipate the cell response, and it's better for targeting metabolism. So I'm just giving an example here on the left. So let's say these the specific cancer type you're studying is very dependent on glycolysis, then you would come with a glycolytic inhibitor. But then what the cell would do is over time, it would shift and it would start using OXPHOS to make ATP. So if you know that, and you can do the opposite. If you have a cancer type that's very dependent on OXPHOS at baseline, if you come with a mitochondrial inhibitor, then it's gonna start using glycolysis for ATP. So an effective strategy would be to come with both pathways at the same time. So knowing what's the baseline of that cancer, you can anticipate the response, and then you can better tailor your therapeutic intervention. So you could do the same for, a global regulator of cancer growth and metabolic drug. Or for example, if you use chemotherapeutics, cells spend a lot of ATP trying to repair the damage caused by those chemotherapeutics. So combining chemotherapeutic and metabolic drug could be a good way to induce cell death in cancer cell. So of course, I want to thank everybody involved in that work. So the first part of the work was led by Sean McGuirk with the help of Yannick Audet-Delage, who's a postdoc in my lab. And the second part of the talk was led by Yannick. We're finishing that story with the help of Catherine, my research associate, as well as Lucia, another postdoc in the lab. We have precious collaborator, the Siegel Lab, Vincent Giguère's lab, and of course we could not do that work without metabolomic core facilities at the University of Ottawa. We had Dr. David Patton and Chamonaz who help us greatly, as well as funding, and I'll be happy to take any question.
Thank you, Julie, for that very exciting talk. We do have a question here from the audience. How might you leverage your improved understanding of cancer vulnerabilities to the development of biomarkers for breast cancer treatment in patient stratification?
Yeah, you could definitely use that for, identifying or stratifying patient. In terms of metabolism, if you look specifically in terms of breast cancer, so it's known if you look, for example, like luminal A and B, which have better outcome compared to, let's say, HER2 positive and triple negative breast cancer, they're different metabolically. So HER2 is very glycolytic. So you could use any marker of glycolysis. You would see that it's highly upregulated. If you take triple negative breast cancer, it's a cancer type that's much more active metabolically, both in terms of glycolysis and oxphos. So, this is one way you could go after that. Like I have some other project, like I did not discuss today, but for example, like cells that have, or breast cancer type that have high level of PGC1 appears to be more sensitive to methotrexate. So we're also doing work to see, for example, PGC1, which is the marker of metabolic state in numerous tissue. I mean, separate from cancer, it's very important for the heart, for the brain, for muscle. Mm-hmm. When you have high level of PGC1, you're predicted to respond better to methotrexate, at least from, preclinical model. So this is one other way you could, use this knowledge of metabolism to inform or potentially inform of, treatment options.
Okay. And this question is more for our trainees. How would you take your findings now into sort of an animal model and looking at resistance and how to target that?
Yeah, so we've done that, like for the first part of the talk, as I've shown, like, so we've taken that in animal models. So where, like in the same animal, we had like a doxorubicin resistant tumor and an epirubicin resistant tumor. And we could show that if you treat the mice with BSO, which interfere with glutathione metabolism, the doxorubicin resistant tumor has harder time growing than the epirubicin resistant one. For the second part of the talk is really, it was really to understand, like, when you have a solid tumor, like not all the cells will be the same. Like, the cells that are on the outer edge, they have access to more nutrient, more oxygen, and they're exposed to higher concentration of drugs. And those in the middle are very different. So our approach was really to try to model that using science, understand, and then, it's interesting. You could look at all the diversity and focus on how they're different. But of course, therapeutically, what we want to do is we want to be able to eradicate all these cells. So then we refocus our analysis, like even though the differences are certainly interesting, we try to find the commonalities between all these cells so that we can intervene therapeutically. And that's why we focus on, the targets we've identified. And then the idea would be that, for some of the target I've shown, BRWD1 and OG and SRCN1, there's no drug available to really target these, but you could do knockdown or stable knockdown. Mm-hmm. And do that in vivo.
Yeah. Okay, well, thank you again. And we really enjoyed the talk, and thank you for being here with us. If there's any other follow-up, we'll get directly in touch with you.
Thank you.
Great.